So in this video, we're going to have a bit of a think about plasmids. Okay, so plasmids are really important when we're thinking about doing any sort of genetic manipulation or biotechnology work, because plasmids are what we refer to as cloning vectors. So plasmid is able to move bits of DNA around. So cloning is where we get bits of DNA from different species and we sort of stick them together or whatever. And a vector is something that can move the DNA around. So for example, a mosquito is a vector for malaria. So the mosquito moves the malaria parasite around. The cloning vector moves the DNA around. Okay, so plasmids are really useful vectors. So they are circular DNA. They come from bacteria. So bacteria naturally have plasmids. So bacteria have both a chromosome that's got all of their essential genes on it, and then they've got these smaller plasmids which have kind of got optional genes on them. So they might have antibiotic resistance, or they might have the ability to metabolize a weird molecule. So they're naturally found in bacteria, but we've kind of exploited them in the lab for genetic manipulation. They are self-replicating in bacteria. So the bacteria has all of the enzymes it needs to recognise the plasmid and to make new copies of it. So they're self-replicating. And they're typically, the smallest plasmids are about 1,000 base pairs up to about 20,000 base pairs. Which is good because a typical gene is 2, 3, 4 kilobases long. So that's 2,000, 3,000, 4,000 bases. So that's a good size that can sort of add into a plasmid if we want to manipulate it around. It's not too big. Once you get above about 20,000 base pairs, it gets more difficult for your plasmids to be replicated, but that's absolutely fine. So what's the workflow? So the cloning workflow, so there is a whole other video on restriction cloning that talks about some of this in more detail, but just to summarise, okay, basically what we have is we start off with a plasmid. So let's say that's 4 kilobases. And then we have our gene of interest, which we've amplified by PCR. So GOI is gene of interest. And let's say that's 2 kilobases. Okay. So what we want to do is, so this gene of interest, we've copied it out of one species. So let's say we've copied it out of one species of plant. And what we want to do is to add it into our plasmid. So what we do is we digest with restriction enzymes. So basically what we do is we cut open that plasmid. And there's a whole other video about restriction enzymes, and there's one on restriction cloning that you want to have a look at for more detail. But basically, we cut the plasmid open. We also cut the ends of this PCR product with the same enzymes. Then what we do is and we have to purify those. We can't just go straight forward. We have to purify out. Then we stick them back together, which is a reaction called ligation, catalysed by DNA ligase. So we've then got our plasmid with the insert in it. So we had 4 kilobases to start with, plus 2 kilobases, so we've now got a 6 kilobase plasmid. Okay, and then what we do is to transform into a cell, usually E. coli, but you can use other things. So then here's my cell. So there's some flagella on my bacterium. Okay, there's the bacterial chromosome that's got all of its essential genes, but we can put that plasmid into the cell, and then because it's self-replicating, that bacterium will start to make more copies of it. Okay. And we can then re-extract the plasmid if we want to. We can do different things with the bacterium, but basically this is the workflow. We have a plasmid, we have a gene of interest that we've amplified by PCR, cut them both open, stick them back together, and transform them into a cell. So typically E. coli is what we usually use, but it depends on your experiments as to exactly what you want to do. So let's have a think about what's in the plasmid that allows this to happen. Okay, so we always draw plasmids as a circle, and in the middle you'll have the name of the plasmid, and you'll usually have the size in base pairs or in kilobases. So nearly all plasmid maps you'll see you've got the name of the plasmid and its total size, because its total size is quite a useful piece of information. So what's the first thing we need? The first thing we need is, it's abbreviated to ORI, so the first thing we need is called the Origin of Replication. ORI. Okay, so this is a sequence that allows for DNA replication. A cell can't start replicating any old random bit of DNA. It needs to know that it's the right piece of DNA. So there's particular sequences that bacteria can recognise, go, ha-ha, I need to replicate that bit of DNA, and then all the polymerases and everything will come in and then we'll replicate it. So if you've got the right sequence in your plasmid, that's where you get that self-replicating behaviour comes from the origin of replication. Okay. It controls the copy number. So some origins of replication, you might only have 2 or 3 copies per plasmid. 2 or 3 copies per cell. Typically, so one of the most common origins of replication is something called PMB. Gives you 20 copies per cell. Some of them allow for hundreds of copies per cell. It depends what you're doing as to how many copies that you want in there. So, but typically you'd be sort of, I don't know, 20, 30 copies. Okay. And then the other thing we need to just bear in mind is if you've got more than one plasmid, so sometimes we have 2 plasmids in E. coli at the same time, use different ORI sequences. So because the ORI is where all the DNA replication machinery will come in and join, if they've got the same origin of replication, then they're competing with each other. So you might not get them both copied properly. So if you're using more than one plasmid, then you'd put different origins of replication in the plasmid so that they were being replicated independently. Okay. Right, what's the next thing we need? The next thing that we need, put it up here. Okay. The next thing that we need is a selectable marker. Okay, so this, a selectable marker is something that you've got a visible phenotype to identify cells with the plasmid. Okay, so this process of transformation down here is pretty inefficient. You typically only get 1 in 10,000 cells takes up the plasmid. Okay, so if you were relying on using PCR or extracting the DNA to identify whether the plasmid has gone in or not, you then have to do 10,000 DNA extractions, 10,000 PCRs. That would be really annoying and really inefficient. So what selectable markers do is they give us a clearly visible phenotype to identify the cells that have got the plasmid. So, for example, we've got antibiotic resistance is the most common selectable marker. There are others out there, but we'll just think about antibiotic resistance. So e.g. you might have kanamycin resistance or ampicillin. Okay, so there are genes that encode enzymes that break down commonly used antibiotics. Okay, so if we use, for example, kanamycin resistance, so let's put CAN-R in there. Okay, we can then tell which plasmids or which cells have got the plasmid and which ones haven't. Okay, so if we think about some agar plates, so let's think about if we've got just standard nutrient agar. in a Petri dish, or if you've got selective agar, so we've got kanamycin in the agar, so we've added an antibiotic to the agar, okay, then that's going to allow us to tell which cells are which. So I'm going to divide my plates up. So on the left-hand side is cells with no plasmid. And on the right-hand side of both plates is cells with the plasmid. Okay. So if we plate our cells, so we do the transformation reaction, but we don't actually give them any plasmid. If we put them on normal agar, then those cells will be able to grow absolutely fine, okay? And the ones on the right, which have the plasmid, will also grow absolutely fine, okay? So, both are, perfectly viable cells. But if we want to find out which ones have got the plasmid inside them, then we grow them on the selective agar. So on the left-hand side, the cells that have got no plasmid have no resistance to kanamycin. So they can't break down kanamycin, so they won't be able to grow. Okay, so we should have a clear plate on the left-hand side. On the right-hand side, we've got the cells that have got the plasmid, and that plasmid contains an antibiotic resistance gene. So they make an enzyme that can break down kanamycin. That means that they could grow on the agar. So what you should find is that you get individual colonies growing on the selective agar, where for the cells that have got the plasmid, okay, so they have got the plasmid in there, they should grow as individual colonies rather than this smear, because only 1 in 10,000 has actually got the plasmid, but then you've got individual colonies, so you might have 20 transformants. You can then go and work with those 20 colonies rather than work with 10,000 colonies. So a selectable marker gene allows you to pick out the ones that have got the plasmid very quickly and easily, and you can tell whether your transformations worked or not. Okay, so each one of those colonies comes from one cell with the plasmid. Okay, so it's got an origin of replication, and it's got resistance to the kanamycin that's in the Petri dish. Okay. So the other big thing that we need in our plasmid, if we're going to use it for cloning or for biotechnology, is to have a multiple cloning site. And I've got other videos, so if you look at my restriction cloning video, it goes through multiple cloning sites in more detail, but I'll do a summary of them there. So we've got multiple cloning site. Which is abbreviated to MCS. Okay, and that has multiple restriction sites to allow for this process over here for digestion and insertion of our gene of interest. Okay. So you might have, so here I'm just going to draw my plasmid out again. Okay, so I might have multiple different restriction sites. So I could have Echo R1, I could have BAM H1, I could have ZO1, I could have SAL1, I could have Hindi3. There's loads and loads of different restriction enzymes, and they've even all got a different recognition sequence. As I say, there's a whole video on restriction enzymes. Okay. What we can do is then cut up the plasmid. So I might digest it with, let's say I'll digest it with Echo R1 and SAL1. And my other video talks about how you choose which restriction enzymes to use, but I can cut that open, which is the digestion process. And then if my gene of interest also has those restriction sites at the end of it, so this is my gene of interest, and I add echo R1 and SAL1 sites, and the other video talks you about how you do that. Okay, if I digest with echo R1, then we'll make the sticky end there. So we've got echo R1 and echo R1. Those have the same sticky end, so those are able to then stick back together. So echo R1 will stick to the echo R1 site in the gene of interest, and the same at the other end with the SAL1. Okay, so when I digest them and then ligate them together, then I can get my gene of interest into the plasmid, okay, and then I can go and select it on there. And what's really good about this is if you use two different enzymes, then what you should find is that different enzymes, you will get positive transformants because in the ligation reaction, the plasmid and the gene of interest stick to each other, so we get a circular plasmid, okay, because we've got two, I'm just going to call them A there and B. We've got two different restriction enzymes, so in this case A is echo R1, B is SAL1. What can't happen if you've got two different restrictions enzymes is that plasmid cannot stick together. So if you've got two different restriction sites, they can't stick together. So echo R1 can't stick to SAL1. This can't happen. Okay, so that means that only your colonies with your insert in will be able to be selected, okay, because the ones with the insert, echo R1 stuck to echo R1 at this end, SAL1 stuck to SAL1 at this end, so we've got a complete circular plasmid that can be replicated. But the plasmid itself, once you've digested it, if you use two different enzymes, then the plasmid with no insert can't religate, so you'll get no transformants. If you just digested it with one enzyme, then it would be able to stick itself back together. But if you do it with two different ones, so you've got echo R1 on one end, SAL at the other end, they can't stick together. So you can't get the plasmids sticking back together. The only complete plasmids will be the one that have your gene of interest in it. So as I say, there's a whole video on restriction enzymes, and there's another one on restriction cloning of kind of how you do this, how you add an echo R1 site, how you add a SAL1 site. So have a look at that as well. But hopefully that's given you an overview of plasmids. Plasmids, really great workhorses. We use them all the time in the lab. Fundamentally what you need in a plasmid, the minimum that you need is an origin of replication. We need a selectable marker so we know which cells have got the plasmid, and then you have a multiple cloning site that you put in your gene of interest in there. Now your plasmids can get much more complicated than that. You can have multiple different genes in there, you can put promoters in there and all sorts of different things that other videos will talk about, but your bare minimum is this system with origin of replication, a selectable marker gene and a multiple cloning sites so that you can put in whatever you like. Okay. The other thing that you might want in your plasmid is we sometimes put in, before the multiple cloning site, we put in a promoter sequence. So rather than relying on copying the promoter of the gene of interest, we sometimes put in an artificial promoter. So we might put in something that's always switched on, which is known as a constitutive promoter. We might put in one that's regulated by temperature, one that's regulated by sugar concentration. So what we often do is to put in a synthetic promoter is in the plasmid, and then we just put the gene sequence into the multiple cloning site. So then we've taken the gene, we've uncoupled it from its natural promoter, and we've put a synthetic promoter in there. So that is optional, but it's in quite a lot of plasmid designs as well. So that can be another feature that you might find in your promoter, and in which case you need to make sure that your gene went in the right way. So in this case, the promoter is going this way, so we need to make sure that our gene was going into, was reading in that direction around the plasmid, not the other direction. So hopefully that's given you an overview of what you need to have a plasmid that you can use in biotechnology or cloning.